Hi, everybody. My name is Charlotte Minio. I'm a student researcher at Union College, and I'm going to talk to you today about how scientists can study enzymes to help make new medications that will allow us to treat fungal diseases. So most people have heard of athlete's foot, or maybe if they haven't had it, they've at least known someone who's had it. And it's pretty gross. It's uncomfortable. It's not something anybody wants. So you go to the store and you buy a cream with an antifungal in it, and you use it to treat your fungal disease. But there are some diseases that are much more complicated and much more life-threatening. And so we need much more advanced and powerful antifungals in order to treat those diseases. But unfortunately, because antifungals are being used more often, they're also becoming less effective. Lots of people have heard of antibiotic resistance. So we use too many antibiotics, and over time, bacterial populations develop resistance to those antibiotics, and the antibiotics can no longer be used to treat the disease. So the same thing can happen with fungi. When we use too many antifungal medications, eventually the fungal populations develop resistance, and the antifungals don't work anymore. So what our lab and a couple of other different labs are trying to do is study enzymes called metacaspases. And what these enzymes do is they're proteins, and it's their job to break apart other proteins in the cell. And pretty much this sends a signal to the fungal cell saying, all right, fungal cell, it's time. It is time for you to die. It's time for programmed cell death. And so this is happening every now and then in the fungi to help them stay healthy. But what we'd like to do is turn on the metacaspases all the time so that all of those fungal cells are getting this message that it's time to die. So in order to do that, we first have to understand what's turning on the metacaspases. So the metacaspases are a protein. So they have this funky 3D structure. And they're sort of like sunglasses. So sunglasses, they have the lenses and they have the side frames that hook over your ears. And when those side frames are bent over, the sunglasses don't really work. They're not going to sit on your face. But something is turning the metacaspases on. It's opening up those side frames so that the sunglasses work, so that the metacaspases send their signal and the cells ultimately die. So we're trying to figure out if calcium can open up those side frames, turn on the metacaspases, and therefore cause programmed cell death and allow us to use metacaspase activation as a new kind of antifungal drug. So in general, what we're doing is studying an enzyme to try to find a new way to target fungal diseases. And the big takeaway from this talk is that if we don't study enzymes now and get this basic fundamental research down, we aren't going to have the knowledge base that we need to develop new antifungals in the future and eventually help patients when they really need it. So thank you so much for listening to my talk. Thank you also to my advisor, Professor Kristen Fox, and the Arnold and Mabel Beckman Foundation for their funding. And I'd be happy to answer any questions. All right, I'm good to stop recording.